Thank you for the nice introduction. Good morning, good afternoon, depends on where you are online. So my name is Yang Liu. I'm from the Division of Bioequivalence 3, Office of Bioequivalence, OGD. So it is my great pleasure to be here to attend this forum and to share some thoughts from OGD regarding the pediatric excipient evaluation from a BE perspective. So hopefully we can all learn something from what we have researched and learned internally, and we would love to share that with the industry. So hopefully by the end of my presentation, the audience can grab some take-home messages or some learning objectives. The first one is to understand the rationale and importance of excipient safety evaluation in pediatric population for generics. And also, hopefully, the industry can have a better understanding of how the OGD's current approach is for the evaluation of excipient in pediatric population when we're reviewing an ANDA. So this is my disclaimer. This presentation reflects the views of the presenter and should not be construed as a representative of FDA's view or policies. So first, let's talk about excipient safety. So I would like to start this talk by sharing, if not the most important, but definitely one of the most critical landmarks in the history of drug and food regulation is the FD&C Act 1938. Actually, the passing of these sets of laws is actually a result of a tragedy. It's known as the 1937 elixir sulfanilamide disaster or incidents. On the left side, I show the drug. That was the original bottle back almost 100 years ago, and also the active ingredient, sulfanilamide, in it. So the drug, sulfanilamide, was an antibiotic. We don't use that often today, but think about that. That's 1930s, and by the time penicillin has not been purified. So that has been a success, and it cured a lot of patients. And there was a increasing demand for a liquid form of this antibiotic. So one of the drug manufacturers, they decided to go for it and to manufacture a liquid-based sulfanilamide. However, in the drug, so the disaster actually is not caused by this drug itself. It's actually caused by an excipient, an untested excipient added into the into the liquid-based drug. And that tragedy, and that response for this tragedy, the excipient, untested excipient, is called diethylene glycol. And that tragedy actually caused at least 107 deaths. Just remember, 100 years ago, we do not have social media, we do not have smartphones, so this is the very conservative death toll, and the actual number could be higher. And within these victims, mostly are children, unfortunately. So this actually brings to a deeper thinking about how we should evaluate the safety of a drug, not only the safety of the drug itself, but also something we put in the drug formulations. From this tragedy, a very important lesson we learned is not all excipients are inert substance. Some may have, and even higher, very high potential toxicity. In general, at this time as we are talking, if an applicant wants to develop a drug, no matter it's a new drug or a generic drug, the applicant must identify and characterize excipient, not only the API, but also the excipient in the proposed drug formulation, and provide information to demonstrate that the excipients do not affect or interfere with the efficacy and the safety. More things are actually, this is not just recommendation or guidance, this is actually law. And if you are interested in more of these, this is where you can find additional information. So at the FDA, as I said in this previous elixir tragedy, most of the victims are pediatric. That's because they are more vulnerable comparing to adults. So FDA generally consider pediatric population to include those patients from birth to younger than 17 years old, or we can say from birth to 16 years of age. And sometimes we do further characterize them into subgroups, as I listed here, depending on their age. And that sometimes is very important for either the efficacy evaluation and the safety evaluation, including what I'm about to talk today. So you are more than welcome to visit the FDA Pediatric website to get more information if you're interested. So the reason that examining the safety or potential toxicity of excipient in pediatric patients is that critical is because the excipient are commonly, the excipient commonly used and determined safe in grown-ups are not necessarily safe in pediatric population. They may have, significant toxicity or toxicological risks. So there is an FDA drafted guidance published in 2019. Actually, is for the industry to understand how the excipient can be evaluated using an inactive ingredient database, or IID. However, at this moment, the IID, it does not provide enough information regarding different exposure models, including safety in pediatric population or other specific subpopulations. On this slide, I'm showing like a list of known excipient that may have safety concerns. I just want to point out it's very hard to read from here, but there are a lot of literatures online that we can find. And what I want to point out is this list is not exclusive, and it's not like only excipient on this list may have a risk, a potential risk for pediatric population. And believe it or not, I'm pretty sure that as we are gathering more and more toxicology data, this list can only grow longer, not shrinking. So in general, in ANDA applications, the recommended BE study, usually we do evaluate the safety. However, even though the drug can be given to pediatric patients, the recommended BE studies usually only recruit or enroll grown-up patients due to ethical or recruiting challenges. So additional pediatric excipient safety evaluation is very important and is very needed, and that's our current practice at the OGD. So before I jump into the next part, I would like to give my first challenging question or check mark. If the maximum daily intake of an excipient in the proposed formulation is below or within the limit of the same excipient for per FDA's IID, it will be deemed safe and acceptable for all ages, including adult and pediatric. Thank you very much. Yeah, it is false because, as I said, the IID is not that comprehensive and it does not reflect the potential risk and potential acceptability for subgroup, including pediatrics. So my next part will be, how we actually do the pediatric excipient evaluation in the OGD. Oh, okay, so let's just jump into I'm sorry about that, the formatting, I think. Let's jump into the second bullet point. So at the OGD, when we're reviewing ANDA and we're reviewing the formulation data, actually all excipient will be evaluated for pediatric population if the RLD can be given to those younger patients. And our first step is usually to compare the excipient levels in the test formulation with the same excipient in the RLD or the approved or approved generic versions of the same product. The reason is the RLD and the approved generics, they will have identical target patient population, identical route of administration and recommended dose, etc. So that is the most conservative way to do the pediatric excipient evaluation. However, if the excipient level in the test formulation cannot be justified by RLD or the approved generics, it will be, it's not like, there's a red light, we do extra steps. It will be compared with other approved drugs for the same route of administration. And in addition, an applicant can submit additional supporting evidence, including toxicology data, literatures, regulatory history, etc., for supporting their conclusion that the use of a certain excipient at a certain level is safe and is not causing concerns in pediatric population. So this kind of work is not only done within the Office of Bioequivalence or divisions of bioequivalence. Is usually involved more offices and sometimes super offices, including the bio team, the clinical team, toxicity team, and also product quality team. And I do want to point out the evaluation of pediatric excipient safety is based on the maximum daily dose. So it's not based on the per unit dose, it's based on how much a patient may take during a day. And that will be taking into consideration of the MDD, maximum daily dose, for different drugs. So pediatric drugs usually have recommended dose for different age groups, and sometimes we do, and need to do further evaluation depending on what age of patients will take this drug specifically. But generally speaking, there is what we call kind of a risk-based approach. That is to evaluate the safety of an excipient in the youngest age group per ROD label. However, this needs to be justified to see that the same excipient will cause the most concern in the youngest group. The rationale behind this risk-based approach is because the youngest age group is usually the most vulnerable group. They have the least developed physiological immunity, and they have the most immature physiological condition or development, such as metabolic system, the GI development, renal development, etc. There is a potential higher risk in the younger pediatrics for the same excipient comparing to older pediatrics or adults. We also need to consider, especially for some older ROD products that has been approved like years ago, the swallowability. Like per FDA's current thinking, we determined that patients younger than 6 years of age will have a potential challenge to swallow a solid base or tablets or a capsule intact. So we also take that into consideration. So let me use a case example to show you how we are actually seeing this evaluation, on how we are doing this evaluation. Let's just assume this is based on a real case, I need to point out, but for confidentiality reason, I just kind of use some dummy numbers, but the excipient names are actually from the real case. I just point out 4 excipients in this example. And as we can see, the first one, microcrystalline cellulose, and the third one, magnesium stearate, they are actually below the level in the ROD or an approved generic version. So in this case, as mentioned before, that's the most conservative way, and these 2 excipients will be determined acceptable in the proposed test formulation. However, the other 2, mannitol and BHT, they actually exceed the limit we can find in the ROD or the past generic version, either because they have a higher level or because BHT was not actually found in any of the approved drug for the same product. So we do take the next step. We need to take the next step is compare that with other approved drug for the same context of use. As I said, based on the risk-based evaluation approach, we evaluate this excipient in a newborn. And I can see, as we can see in this slide, mannitol can be justified by other drugs, while we're still having a hard time finding any drug approved before that has a higher BHT level in the group of newborn. So this will raise a concern that whether this can cause safety issue or can cause a toxic effect, has a toxic effect in this subgroup. And very limited information can be found regarding the BHT safety in newborns from the literatures. So we do need to point out, and that's actually what happened, is we actually pointed out to the applicant, asking them to provide additional toxicity data or additional evidence to demonstrate the safety of this particular excipient for a newborn use. So then my next challenge question is, which of the following statement is true? Number one, OGD only evaluate safety of the most riskiest excipient in pediatric population, or OGD evaluated this pediatric excipient safety evaluation based on MDI, or the excipient levels in proposed generic formulation is evaluated only by comparing to the RODs. And the answer will be the second one. The problem with the first one is we're not only evaluating the riskiest excipient, we actually evaluate all excipient if the drug can be given to pediatric population. And the problem with the third is, yes, we do compare that with ROD and other approved generic, but that's not all. If these drugs cannot cover our test formulation, we do take extra steps. There are a couple of extra considerations when we are doing this pediatric excipient evaluation. When the drug or when the proposed excipient level cannot be justified by ROD or approved generics. So the first consideration is whatever drug we are trying to use to justify the current formulation, it should be given to same or even younger age groups based on the rationale I provided before. And also the drug we want to reference or we want to use as a justification, it should be available on the market, or at the bottom, at least it should be discontinued, not due to safety or effective reasons. And the context of use during the excipient evaluation is very, very important. And I will spend the next slide talking about that in a little bit detail. I still have 3 minutes, I guess. So the first concern or the first item we check about context of use is the route of administration. At this moment, we cannot accept like a cross route administration justification. So the drug we want to use for justification must have the same route of administration. The second thing we consider is the duration of treatment. That is because the excipient evaluation we do is based on MDI, is based on the daily intake. However, the drug can be taken for a different amount of periods. So some are taken for a short term, some for chronic use, etc. An example would be if, saying we're reviewing a drug for supplemental hormone medication, and we want to justify one of the excipient. Probably we do not want to use a PET imaging agent, which you just take it once. So this consideration is based on potential risk due to long-term intake. We also consider the severity of indicated diseases between the drug we want to justify and the drug we want to use for justification. An example is if we are actually trying to justify excipient in a formulation of an anti-coughing syrup, probably it's not the best idea to refer to a chemotherapy formulation because the patient will take different kinds of risks. And also swallowability of a solid dose form, if the LD specified actually make it very clear that the drug should not be chewed or should not be crushed before taken. If that's the case, we do need to take into swallowability issue in younger pediatrics, for example, younger than 6 years of age. So I can't help showing this picture. This picture was actually cited from my dear colleague from the OGD. Don't worry, this is not a real picture. This is my colleague generating this picture using AI. So this just reflects our thought that we do want to take consideration of as much as possible during our excipient evaluation. So this will be my last challenge question. It's a little bit long, but let's say if we can think from an OB reviewer perspective now. So the proposed generic drug in a new ANDA is an orally dosed anti-infectious drug. It can be given to grown-ups and patients older than pediatric patients 2 years of age and older. The recommended dosing regimen is a 4-week treatment, daily treatment. I should have said that. So which of the following if they are all approved, they were all approved in the past, which of them are the best to justify the excipient in current test formulation? I'll just pause here and give everybody like a few seconds to read the options. Okay, I'm going to jump into the answer. The answer is B. Instead of talking more about option B, again, let's see what the problem there are with option A and C. In answer A, it says an anti-inflammatory drug indicated for colitis in adult patients. It has a 3-month recommended dosing period. So this actually is not recommended for pediatric, so it cannot be used to justify pediatric drugs, even though it has a longer recommended dosing treatment period. And for number 3, an anti-seizure drug that can be given to grown-ups and pediatric 1 year and older, but it has a boxed wording for toxicity in the RLD. So this one actually, if we forget about the severity of the disease, this is a acceptable justification. However, if we do consider the severity of the disease these 2 drugs are treating, this is not the best idea. So to wrap up my presentation, there are a lot of thanks I want to give to my dear colleagues. First of all, I want to special thanks to Dr. Partha Roy, the OB director, who's actually presenting here. So Partha actually made the whole thing, multiple projects and multiple working groups available and going in the Office of Bioequivalence, and thank you very much for the support. I do want to especially thank April and Lisa from OB and OSE respectively. They are the pioneers that actually put these projects on table. I want to thank Dr. Ki Ren, and she's Deputy Director of DB3. She has been, along the way, supporting and also leading the working groups, multiple working groups. I just list 2 working groups here: the Pediatric Evaluation Working Group and the internal Pediatric Excipient Database Group for a lot of supports. And last but not least, I want to thank OGD, OB, and OSC management and staff for their great support. And of course, thank you for your attention. I would love to take questions. Please feel free to put it in the chat. Thank you. Now I'm going to turn it to my dear colleague, Tao. Thank you very much.
Good morning, or good afternoon, good evening. My name is Tao. I'm from Office Bioequivalence. I'm Senior Advisor. Today, you can see that what I'm going to talk about is a little bit different from other speakers. But I just want to say, just now you heard my colleague talking about how bioequivalence assessor assesses safety for pediatric use for inactive ingredient in the proposed formulation during ANDA assessment for bioequivalence. Next, I'm going to tell you where this assessment occurs and how efficient this assessment could be by using a modernized regulatory assessment tool called GDSA-BE. We give it a beautiful name, just easy to remember. Listen carefully. It's called GDSA-BE, okay? So hopefully you all can remember that after my talk. But here I'd like to pause and make a quick poll in our on-site audience. So how many of you have heard another agency's structure assessment initiative called CASA? Please raise your hand if you do. Okay. How many of you have heard GDSA-BE before today? Okay, none. Oh, maybe one. All right. So actually, it's not surprising. Today is the first day or first time that OGD is officially talking about this great modernized generic tool, generic assessment tool. And I have the greatest honor to do the job today. So please do me a favor. If you could only remember one thing from my talk, that would be OGD is scrapping text-based, decades-long review process for GDSA-BE. This is a modernized generic regulatory assessment for accelerated bioequivalence regulatory process, right? For accelerated review, for efficient review, for streamlined review. So we're changing for good. Okay, now let's see. So why we are initiating this GDSA-BE? Any thoughts? Because we need a modernization, right? For decades long, we have been sort of thinking about what we should change for this text-based review process. We know we need a change, but just recently, we have, every day we see these new innovations such as AI, ChatGPT, right, Sora, continue influencing humanity for almost every industry in the outside world. That makes us think and make us cannot stand still with the current text-based review. We just need a change. Thanks to the IT technology advancement, that makes this possible for us. Now we come up with this modernized generic drug assessment called GDSA-BE, and this is really the next generation of generic drug assessment process for bioequivalence. And please remember these 4 words: consistency, standardization, efficiency, and knowledge management. These words are really the model of GDSA-BE. So these are really, highlight the benefits this GDSA-BE tool can provide to the entire generic drug program. And the need for the modernization also comes from the challenges, right? The challenges that we're facing for the current regulatory assessment process. For example, externally, growing complexities with ANDA submission that makes us feel we need a streamlined assessment process in order to successfully meet the GDUFA goal dates. And also the growing expectations from Congress, from the public, from all of you have motivated us to make the change. Like I said before, the current regulatory assessment is not ideal, not perfect, right? We recognize there are a lot of room to improve this process. So the freestyle narrative text-based review just provides a lot of limitations. And for example, knowledge management, it just doesn't give us a very good visibility, right, for any previously completed review. Also, it involves a lot of manual administrative tasks. For example, reviewers need to copy pasting data from the submission. Reviewers need to manually searching databases in FDA data sources, right? So all of these need improvements. And most importantly, we feel during the development of GDSA-BE, we feel we probably also need structured data submission from ANDA applicants, okay? So think about, right, in the future, if we have structured data submission, we can make this interface with GDSA-BE. So GDSA-BE can really achieve a very high level of automation, right? And this will really help free our assessors from the manual excessive work and allow them to focus on the areas that really require human intelligence and their professional expertise. So now we have talked about why we made the change. Really, the modernization is a key, right? And actually, modernizing the generic drug program is OGD's top priority for its 5-year strategic plan between 2023 and 2028. So for the process, we'd like to modernize to enhance efficiency and consistency across the entire generic drug program. And we believe that once GDSA-BE is fully developed and implemented in OGD, and we can really enrich the effectiveness and consistency for the regulatory assessment for bioequivalence. And really, this will be a significant step for OGD to make, right, towards achieving this goal, modernizing the generic drug program. And OGD has published 2023 annual report. It is available online. So we have shared a little bit details about the progress we have made to developing this tool in 2023. If you are interested, please download a copy from the internet and take a read. So at this point, I believe everyone probably understand what GDSA is, right? Generic Drug Structure Assessment. So this is really symbolizing the next generation OGD's regulatory assessment tool for for generic drugs. So this is kind of busy slide, but I'd like you to focus on these 4 graphics on the slide. So let's take a look at the middle row first. This illustration really help you to understand from the old or decades-long text-based disorder form, which is on the left, to move to a more future structured state of the regulatory assessment. And this future state is much more ordered, much more structured, and it has this dynamic interactive capabilities with all the different resources. I'll explain that in a little bit of detail in next slide. So with that thinking, the bottom 2 images will show you an example of GDSA-BE using generic drug regulatory bioequivalence assessment as example, right? The bottom 2 images will let you know the comparison view of regulatory bioequivalence assessment before GDSA-BE, which is the left image, and after GDSA-BE, which is on the right bottom corner. So before GDSA-BE, the bottom left image is a snapshot of our of our regulatory assessment for bioequivalence. You can see all of these rows and columns needs to be manually filled by our assessors. And they'll need to go to the submission and manually retrieve the data and put that in this review template. Okay, and now once we move to the GDSA-BE stage, and this is the interface of GDSA-BE, by the way, and they only need to enter this and the number, and then a lot of information from the left image will be auto-populated for them. Okay, think about that. And in this red box, it really highlights the future state of generic drug regulatory assessment. So how efficient, how streamlined it could be once GDSA is fully implemented in OGD. Okay, so now I come to my $1 million slide. So if you could only remember one slide from my talk today, this would be it, okay? So as I said, GDSA has a model, right? The 4 words: efficiency, consistency, standardization, and knowledge management. So on this slide, it really tells you why the features of GDSA-BE can provide generic drug program all of these benefits. So first, data automation analysis. So GDSA is a tool that we could integrate this tool with some assessment tools that we developed in-house. And by using these assessment tools, currently, we are able to achieve certain level of automation. For example, we can auto-populating some data from the end of submission to GDSA-BE. We can even auto-analyze some PK data using that tools. And we're visioning, right, in the future, once the structured data submission is in place, we can achieve even more high-level automations. So this really helps cut down the time that reviewers spend in manual administrative tasks like copy pasting and allow for a higher efficiency for the review process. Second, data mapping and comparison. So remember I mentioned in the previous slide that GDSA-BE is a tool that we could have more interactive capabilities with various data sources, right? These data sources in FDA resource page that we could create a linkage with GDSA-BE. So GDSA-BE can link to these data sources, and previously, assessors really need to go to these data sources one by one, and some of them may be in the different systems. They have to go jump in and jump out of the system to do a manual search of the data that they need to complete their review. But once we link GDSA-BE with these data sources, the assessors really just need to sit in within GDSA-BE, right? Once they opened up GDSA-BE, all of these data sources are available to them. They don't need to go in, different systems to find the data. Just one click away, they can have the data available to them, or even auto-imported from these data sources to GDSA-BE. So think about how efficient that process could be, right? We are saving tremendous amount of time for the reviewer spend searching databases, searching data sources, and we also allow a higher level of standardization and consistency for our review process. And third, we can also link GDSA-BE with various different language libraries. So over the years, right, based on all the years' review experiences, we have developed all different types of language libraries. And we can really link GDSA-BE with these language libraries, so we can make sure a consistent deficiency language can be used for similarly situated cases. Right? And also we can capture any new unique deficiency language that's used for a unique scientific issue. So in that case, this new deficiency language can be used for the future use, right? So this really helps me, it's a great segue, help me to go down to my last, but not the least, probably the most prominent feature of GDSA-BE, which is knowledge management. Okay, so at this point, I hope you probably get a sense GDSA-BE can capture data, store the data, curate the data, and use that data for future cases, right? So this is really a great knowledge management tool, and also, intelligent bioequivalence review tool. Really. So this is a slide I really want everyone to remember in your mind when you walk away from today's presentation. So GDSA-BE is our next generation regulatory assessment for bioequivalence, and it is both an intelligent review tool and a valuable knowledge management tool for Office of Bioequivalence and OGD. Lastly, I want to share with you a little bit about where we stand today for GDSA-BE. Since 2022, we have launched 3 releases, including the very first software release in December 2022, and there are about 60 applications reviewed in GDSA-BE system now. And I want to emphasize that GDSA-BE is fully implemented in Office Bioequivalence. And with that, it's my challenge question time. GDSA-BE, Generic Drug Structure Assessment Bioequivalence Tool, is A, Generic Drug Bioequivalence Review Tool, B, Bioequivalence Knowledge Management Tool, and C, both. Any takers? C, right? Yeah, it's obvious. And in summary, generic drug bioequivalence assessment is entering a new era. Right? You all can see that. We are dumping the old text-based review. We are very excitedly going to this new stage, GDSA-BE. And like said many times, this is not only an assessment tool, but also a knowledge management tool. And with GDSA-BE, we are looking to achieving higher efficiency, higher standardization, and we can empower our assessment to make more consistent and more informed regulatory decision. And so this is a wonderful tool that the entire Office of Bioequivalence and OGD are very excited to embrace. So with that, I end my presentation today. But before I leave the podium, I just want to make another quick poll. By now, how many of you know GDSA-BE? All right, a lot more hands, right? Thank you, and it looks like you learned something new from my talk, and thank you for helping me achieve my mission. And I'm very happy to come back to answer any questions you have in the Q&A. All right, so with that, I'd like to turn it over to my colleague, Andrea and Shebnam, for Bio-IND.
It's It's always tricky to follow a talk about modernization with my paper notes. But then again, I'm also that person that like reads actual books, like really paper books. I'm that old. So, all right, so I'm here with Dr. Ferrugi, and we're going to be talking to you today about Bio-INDs and comparative analyses. 2 for 1 here. All right, bonus deal. Let's ride. Just as a disclaimer, this presentation reflects the views of the authors and should not be construed to represent the FDA's views or policies. So to start things off with Bio-INDs, I'm going to give you some tips here about how to avoid clinical holds and how to just streamline your Bio-IND processes. So I'm Andrea Dugas. I'm a reviewer in the Division of Clinical Safety and Surveillance. I am a physician, and we are the division that is responsible for reviewing these Bio-INDs from a clinical safety standpoint. Obviously, these are going to be going over to our colleagues in bioequivalence and chemistry and all that, so what I'm going to talk about here really is focused on clinical safety. So there are 2 things that I want you to take away. She just had 1 thing. I've got 2. You guys can do it. I have very high expectations for you. 1 is to be able to identify the FDA resources that are there to assist the Bio-IND process. Hint, they're at the end of this presentation in the resources slide. It's like labeled. And second is to learn some tips on how to avoid clinical holds and save time and back and forth on these Bio-IND submissions. So no FDA talk would be complete without mentioning 21 CFR. This is my obligatory slide. I did it. So as opposed to the Office of New Drugs, where INDs are really going to be a lot of times new to human or not approved drugs, these are already approved. They've already been in humans. So there are 3 reasons why people would need to submit a bioequivalence IND. And that is if the study includes a radioactive product, if it includes a cytotoxic drug product, or if the study has a product in it, and it could be the drug, it could be some other product that is being given above the dose that is recommended on the labeling. And when you submit those Bio-INDs, like I said, we do the clinical safety review in addition to the multiple other colleagues we have at the FDA that do reviews of Bio-INDs. And from our perspective, we can issue a clinical hold, which basically stops the study and does not let it continue, or does not let it begin if it hasn't begun yet. Or we can give you non-hold comments. These are helpful suggestions on kind of how to improve the safety or study design. There are 4 reasons from a clinical safety perspective that we can put that Bio-IND on clinical hold. So the first is if human subjects are going to be exposed to an unreasonable or significant risk. That's actually the most common one. That's the main reason in this day and age that we tend to put studies, Bio-INDs on clinical hold. The other 3, you can fairly easily avoid it with some due diligence. So 1 is if the clinical investigators are not qualified to conduct the Bio-IND. That's rarely the case, but what happens more often is people just forget to submit the CVs of those investigators showing that they are qualified to do that. So that one can be avoided for the most part by submitting an appropriate CV showing that the investigators are qualified. Number 3 is that the investigator brochure is misleading, erroneous, or incomplete. Again, submitting a complete, accurate investigator's brochure, boom, knock that one out. And then the 4th one is that if the Bio-IND doesn't contain sufficient information to demonstrate to assess the risks of safety to the subjects, and again, submit a complete study protocol. So if you submit those 3 things, a clinical investigator CV showing they're qualified, a complete and accurate investigator's brochure, and a study protocol, that's really going to narrow this mostly down to the main reason we put studies on clinical hold is significant risk or injury to the subjects. So we took Bio-INDs that have been reviewed over about 18 months and looked at all of the comments that we gave back to the applicants or sponsors and categorized them into kind of main class categories to see where we are having most of our comments and critiques on these Bio-INDs. And this is a lovely pie chart, even though in my PhD they told us never to use a pie chart. I am a rebel. So as you can see, this little like wedge that's like 8% 90% for application completeness. We already talked about that. That's where you submit the, investigator's brochure, the complete protocol, the investigator CVs, boom, taken care of that one. Then, the biggest wedge there in red is selection of subjects. And that's really where we have the most comments going back to the applicants. And so I'm just going to walk right around that pie circle, and we're going to talk about each one of these topics and give some specific tips on how to improve submissions. So, study design and protocol tips. You're going to hear me repeat this over and over again with every slide. My first 2 points are going to be look at the PSG and look at the RLD label. So the FDA issues these product-specific guidances for generic drug development. They are not mandatory, they are guidances, and you can absolutely use an alternative approach, but I would suggest you to give adequate justification. But these are our best thoughts on how to complete these studies, and so they bear to look at and consider. And then the next is to look at the RLD labeling. That's what we consider too. It really highlights some of the safety issues that are going to be occurring with this drug. So, subject selections. Shockingly, I'm going to say follow the PSG. Who would have expected that? But the PSG is going to give you tips on study design. It will let you know, maybe this is recommended more in patients versus healthy subjects. The RLD labeling is going to let you know who should not be receiving this drug, who has contraindications to it, what are some potential drug interactions. So, if a drug is contraindicated to be given to patients with thyroid disease in the subject selection, we as reviewers are going to be looking, make sure patients with thyroid disease are not, are excluded from that population so that we know that we're keeping them safe. Something that I see a lot of is that, protocols will say that they are enrolling healthy subjects with normal vital signs or with clinically insignificant. That is very vague. It is hard for me as a physician to know what you mean by a clinically insignificant vital sign. And so who's ever enrolling your patients is going to have a hard time too. So that's why we really recommend giving specific parameters. Instead of, normal vital signs, say, normal vital signs, and this is what we consider normal. And the same with lab values. With normal laboratory values, okay, just give us your normal laboratory values so we know what you're talking about. If a drug is contraindicated, say in people with liver disease, then again, I will look to the inclusion-exclusion criteria to make sure that you're only enrolling people with normal liver function. And normal liver function means normal liver function within the range of normal liver function that you gave me, not 5% above that or clinically insignificant, liver values. Study drug administration. Again, the PSG will give you the kind of recommended dose. Sometimes they will comment on timing. In terms of the RLD labeling, they'll also give instructions. If the RLD labeling goes into detail on how a drug should be prepared, dilute it like this and infuse it over this time with or without a filter, I would expect that also to be in protocol so that so that we know that the subjects in the study are getting the drug the way that is recommended be administered when it is given in patients. Also, if a drug can cause anaphylaxis or some sort of severe allergic reaction, it's important for us to know from a patient safety perspective, how are you assessing for that? How are you checking those patients? How often are those subjects? How often are you checking them? Do you have a code card available? Do you have epinephrine available? How are you going to be identifying and monitoring these subjects for anaphylaxis? And then the last thing I'll say is that if you are enrolling patients that are taking this drug for a disease, then for their safety and for their treatment of their disease, they need to stay on their regular treatment schedule. And that treatment schedule should not be changed because of the study. So sometimes we'll see, a drug dose may be delayed for administrative reasons. Well, that's really interfering with the patient's treatment. If the patient needs to stop treatment because of some adverse event, that's very different rather than just for the study. All right, safety monitoring. Again, look at the PSG recommendations, look at the RLD labeling. That will give you a lot of information about the types of adverse reactions that need to be monitored. Again, define what is a normal vital sign range, define what is an acceptable laboratory range. And then consider the timing and the frequency of how you might do that laboratory testing or vital signs or EKGs, et cetera, whatever you're using to monitor these subjects. So if a drug can cause neutropenia about a week after giving it, checking the white blood cell count the next day isn't going to detect that adverse event. That's not going to help protect the subject's safety. You need to check for the neutropenia a week after, and whether that's having them come back for another study visit or combining it with another blood draw. But the timing of it needs to make sense in the context of the adverse reaction that you're looking for. Same will be, if a drug can cause liver injury, then it would make sense to check and make sure that the patient does not have liver injury before continuing to give them additional multiple doses of that. And the one safety monitoring timing that I see actually the most is that, so for drugs with embryo-fetal toxicity, people are testing for pregnancy before each dose. That's great, wonderful, spot on. But also just remember to check for pregnancy at the end of the study, just so that we can, record any sort of adverse events and discuss briefly how you plan to follow that up. All right, subject withdrawal. If a subject develops a contraindication to the drug during the study, maybe that's an allergic reaction, maybe that's a kidney injury, something like that, then they should not be receiving further doses to it. So if they develop a contraindication, that needs to be listed in the subject withdrawal criteria so we know that those subjects will be safely withdrawn and not additionally dosed with the medication. Adverse event reporting. This is actually usually done pretty well. There's a really nice guidance for industry that the FDA put out that talks about safety assessments. The one point in here that I do kind of notice is there are specific adverse event reporting for Bio-INDs. They're going to be submitted for marketing for the FDA, that you need to report those to the FDA, and with a timeline. So if there is a serious and unexpected suspected adverse reaction, that needs to be reported to the FDA within 15 calendar days. And if there is an unexpected fatal or life-threatening suspected adverse reaction, that needs to be reported to the FDA within 7 days. And just make sure that that is also included in your adverse event reporting. Informed consent. Again, the FDA has some nice guidance here on informed consent guidelines. The main thing is just making sure that the informed consent is clear and understandable. If you tell a patient that they could have hepatic toxicity, they probably are going to have no idea what you're talking about, stating as, you could have damage to the liver or liver injury, in a way that is understandable to the subjects. Make sure to fully inform the subjects about the study risks. One tip there is if the label has a medication guide on it, that can be tacked on to an informed consent, and that will walk through at least some of the safety risks related to the drug in a way that patients or subjects can understand. And then also, if you already have a study event table in the protocol, that is something that could be added to informed consent as well, in order to help fully inform the subjects about the study events. And my last point here is just, if there are specific study restrictions that the subjects need to know about, put that in the informed consent. That way they have been clearly informed of them, and we know they've been clearly informed of them. The most common one there would be contraception. If they're required to use contraception during the study and for 4 months afterwards, just make sure that's clearly spelled out in the informed consent, so that that is highlighted for the subjects and they know about that. Everything I've said before is about Bio-IND submissions, mostly about new submissions, but for Bio-IND amendments, just to touch base there, if you are amending the Bio-IND, just include a track changes protocol, and ideally it would be very nice to have like a little table with the differences between the new submitted protocol and the current study protocol. That just highlights for us what the changes are, the rationale for the changes, and really helps us kind of understand the thought process and ensure that there is no change to patient safety with this new amended protocol. So this is that big highlighted resources page I promised at the very beginning. Here's a list of a whole bunch of resources. I have those FDA guidances that talk about informed consent, that talk about adverse event reporting. There's information here about when to submit a Bio-IND or exceptions for a Bio-IND submission, and a reference actually to a presentation here 2 years ago that was really great, that kind of walked through the submission of INDs and when to submit it. All right, challenge question. Which of the following statements about Bio-INDs is not true? I will just let you read through these for a second. And in the interest of time, because Dr. Ferrugi told me that if I went over my time, I have to bake her a cake. And I don't have time today to bake a cake for her. So you're going to lose your cake. I'm going to jump. The answer here is bioequivalence study in healthy subjects absolutely needs to consider the RLDs, labels, warnings, and precautions. That's how we, that's how you're going to make sure that these subjects are not having, significant adverse events and that they are staying safe for this study. So in summary, the FDA has multiple resources to help you with these Bio-IND submissions. I strongly encourage you to come back to this presentation online and go to that resources page to find a large number of them. And just again, refer to the RLD label and to the PSG when you're designing your Bio-IND studies. And with that, and 30 seconds over and a cake I have to make later, I pass stuff over to Dr. Ferrugi.
Thank you, Dr. Yus, for the nice introduction. And to continue on with the clinical theme, my name is Shabnam Ferrugi. I am a clinical reviewer with the Division of Clinical Review within the Office of Safety and Clinical Evaluation, and today I will focus on comparative analysis update. My learning objectives are to review the comparative analysis process, to provide key principles for conducting comparative analyses, and to discuss tips for user interface assessment during product development. Generic drugs are considered to be therapeutically equivalent to the reference drug if they have the same clinical effect and safety profile when administered to the patients under the conditions specified in the labeling. The same expectations are applicable for the generic drug device combination products, and FDA considers whether end users can use a generic combination product when it is substituted for the RLD without the intervention of the healthcare professional or without additional training prior to the use of the generic product. That being said, generic and RLD products do not need to be identical as long as the differences do not preclude approval under an ANDA. The comparative user interface assessment process is a cross-office collaborative CEDAR process. Our groups work together on comparative analysis and comparative use human factor studies throughout the ANDA lifecycle. Note that the guidances for comparative analyses apply to different stages of the lifecycle. They apply in the pre-ANDA and ANDA submissions, but also in the submission of post-approval changes and supplements. Within the DCR, we are the lead for ANDA submission and post-approval CA assessments of combination products to ensure they have the same clinical effect and safety profile as their RLD. This slide provides an overview of the types of combination products that were submitted in ANDAs to OGD. And just like Dr. Dugas, I have a pie chart as well. And my chart represents the CA assessments done by DCR from 2018 to 2023. And as you can tell from the pie chart, injectable and oral products make up over 2/3 of the combination products submitted under the ANDAs. So the draft guidance for industry, published in 2017, provides the current recommended approach to comparative analysis in an ANDA. And I will go over the 3 comparisons in the next few slides, which are the physical comparison, the comparative task analysis, and the labeling comparison. Within each comparison, consider and include all components with which end users interact. Before going any further, let's review some key definitions. User interface, that refers to all components of the product with which a user interacts. This includes the delivery device constituent part and any associated controls, displays, product labeling, and even packaging. A critical task is a user task that if performed incorrectly or not performed at all would or could cause harm to the patient or the user, where harm is also defined to include compromised care. Ultimately, an external critical design attribute is a feature that directly affects how users perform a critical task that is necessary to use or administer the drug product. Let's just start with the physical comparison. In your submission, please include clear, detailed, and color photographs. And according to draft guidance for industry, in your comparison, you should consider the visual, the auditory, the tactile examination of the physical features of the proposed generic to the RLD. such as the size, the shape, the color, the resistance, and even the sound. Include all components necessary to deliver the drug, from packaging to the connectors, and external design mechanisms and features should also be noted. Ultimately, please clearly identify, characterize, and provide justifications for the differences noted. For the comparative task analysis, we recommend that you physically manipulate the device and systematically analyze and compare the sequential activities required for the end users to use the generic drug product and administer the drug product. Be sure to include all the steps with which end users need to perform to use the device, from opening the packaging to disposing of the product, and highlight the differences in tasks that arise due to differences in user interface design. Be sure to note if those differences may impact an existing critical task or even give rise to a new critical task. For the labeling comparison, use the current version of the RLD labeling. Ensure that labeling, including IFU and images, accurately describe your proposed generic combination product. Make sure that all the tasks necessary to use the generic combination product is also identified. Generic drug product labeling generally must be the same as the RLD. However, certain limited exceptions are allowed, and those will be evaluated on a case-by-case basis. Be sure to evaluate labeling statements on the container label and even carton labeling. These may include important elements that are intended to minimize medication errors or use errors. IFU differences may also reflect differences seen in comparative task analysis. According to the draft guidance for industry, for each comparison, you select an outcome, and those outcomes are no difference, minor design difference, and an other design difference. All identified differences should be considered, again, within the context of the overall risk profile of the product. If you choose the outcome of no difference, please provide explanation in the comparative analyses to support your assertion that there are truly no differences in the design of your proposed user interface as compared to the user interface of the RLD. A minor design difference, on the other hand, will not affect an external critical design attribute. For instance, the different color of the plunger rod for a prefilled syringe, and also knowing that the color of the plunger rod is not critical to the correct use of the device. If FDA agrees that the design differences is a minor design difference, then it would likely be acceptable. On the other hand, an other design difference is one that may impact an external critical design attribute that involves administration of the product. When you have other design differences, consider redesigning the user interface to minimize differences from the RLD or provide additional data to support the user interface design difference. The type of information and data will depend on the differences and risks to be considered. With appropriate outcome classification and justification, it is reassuring to know that most deficiencies are communicated and resolved within the review cycle. Indeed, between 2018 and 2023, 90% of the CA assessments that were done by DCR were found to be adequate, and only 10% of them had another design differences that were inadequate. Now let's go over some general recommendations. Identify and provide adequate justification for all the differences in the user interface in your comparative analysis. Focus on potential differences in the critical tasks between the RLD and your generic drug product combination. Always consider the context of use. Let's go over the context of use. For instance, a drug may be intended for an emergency use versus non-emergency use. The frequency of use is also important. Is it intended for single use or repeated use by the end users? The end users may be your lay users who have no experience with the drug. That may be a patient or a caregiver versus an experienced healthcare professional. Ultimately, also consider the environment of use. Is this drug going to be used in a non-clinical setting, such as homes and schools, or in a healthcare setting, such as inpatient hospitals or outpatient clinics? Ultimately, always consider your patient populations, those who suffer from dexterity issues, from a rheumatologic or a neuromuscular disorder. To continue with the tips for your comparative analysis, Know that design differences are product specific and must be analyzed within the context of their comparison to their RLD. If the RLD is discontinued and or unavailable, we highly recommend submitting a controlled correspondence or a pre-ANDA meeting request to discuss the alternative approach with the agency. Use the to-be marketed generic information product in your comparative analysis. Engage with FDA early during your product development via controlled correspondences and pre-ANDA processes. Submit your comparative analyses, the samples of your products, and specific questions in pre-ANDA communications request. And if you identify other design differences, we again highly recommend discussing that early with us. Incorporate recommendations in the draft comparative analysis guidance throughout your combination product, and where able, design the generic product to minimize differences in user interface and critical tasks as compared to its RLD. Perform your comparative analysis throughout the development program, especially if changes are made. Let's talk about the changes. Generally, when there are changes to your proposed product after approval, these are submitted as a post-approval changes and supplements. Again, DCR is the lead for the assessment of the comparative analysis submitted with post-approval changes and supplements. A change may be a change that's applicable to the device constituent part. For instance, if a new oral dosing syringe is needed to replace the approved dropper, or a change from a single-use vial to a prefilled syringe, or a change of the color of the dust cap of the inhalation device to match the RLD. Some of those changes may also apply to the labeling, to revise the graphics and the text of the instructions for use to be consistent with the RLD updates, or to represent the revised needle shield without other device changes. In all those situations, submit a CA report with supplements reflecting changes to the device and device-related labeling, and mention the CA report in your cover letter or submit it in Module 5, which is the preferred module for the submission of those CAs, and compare the updated drug-device combination back to the RLD. Again, when in doubt, submit a CA report with changes and consider a controlled correspondence with us. Now, time for a challenge question number 2, true or false. The physical comparison includes visual, auditory, tactile examination of the physical features of the proposed product compared to the RLD. Of course, the answer is true. So to summarize, please refer to the draft comparative analysis guidance for all the recommendations. Again, note that all design differences should be identified, adequately analyzed, and scientifically justified, and ultimately engage with us early during your combination product development. Thank you very much. This thumbs up for me. I will be back for the Q&A answer. Thank you very much for your attention.
Hello, everyone. My name is Jimena Dancy. I'm a pharmacologist in the Division of Pharmacology, Toxicology Review within the Office of Generic Drugs. And today I will be presenting successful practices for pharmacology, toxicology, justifications, and ANDAs. This is my disclaimer. Here's a brief outline of my talk. First, I'll present the learning objectives, introduce you to PharmTox Review in the Office of Generic Drugs. I'll provide you with some common issues and deficiencies that our division encounters, and provide you with some best practices and resources for submitting PharmTox justifications. Here's the learning objectives we're going to cover today. I will come back to this slide throughout the presentation, but the first one we will go over is understanding the role of PharmTox Review in OGD. Before I go into details, I would like to provide you with a brief overview of the role of the Division of Pharmacology Toxicology Review, or DPTR, in the safety assessment of generic drug products. We conduct safety evaluations on excipients and impurities in generic drug products on a consult basis. And we conduct our reviews within the context of the ANDAs we're consulted on, so we take into consideration the dose, duration of exposure, patient population, and route of administration. Our goal is to determine that the generic drug product has the same safety profile as its RLD. And DPTR is involved at various stages in the life cycle of the generic drug product. In the development phase, we participate in pre-ANDA product development meetings and work on control correspondences. In the pre-marketing phase, we review drug master files or DMFs, ANDAs, and control correspondences. And in the post-marketing phase, we review ANDA supplements and health hazard evaluations. And occasionally, we review citizen petitions and media content. So what does PharmTox and OGD do? We review the safety of generic drug formulations where their safety profile may differ from the RLD. And these differences may be in the form of impurities, excipients, residual solvents, and contaminants from the container closure system. DMF holders and ANDA applicants submit toxicology data to support specification limits when these limits exceed allowable safety thresholds. And these justifications may fall under three main categories. The first one is written justifications, for example, collections of published information or written expert opinion. The second category is in silico prediction. These are mainly in the form of quantitative structure-activity relationship, or QSAR, predictions to determine bacterial mutagenicity. And then lastly, in vitro and in vivo studies, and these are generally to qualify genotoxicity or general toxicity through repeated dose toxicity studies. Next, I will present you with some PharmTox safety assessment challenges. And the first topic we will cover are impurity review in generics. And impurities are any component of the drug substance or drug product that is not the drug substance itself or an excipient of the formulation. Now, please note that generic drug formulations may have different impurity profiles as compared to the RLD. And these may be in the form, drug substance or drug product related impurities, elemental impurities, residual solvents, and extractables and leachables may arise during the manufacture and or storage of the API or final drug product, which is illustrated in the diagram below. And DPTR evaluates the safety of impurities when their specifications exceed the safety thresholds that I mentioned in the previous slide. And I will go into more detail on these thresholds in the coming slides. And there are different approaches that an applicant may take to qualify an impurity that exceeds these allowable safety thresholds. One is a comparative impurity analysis with the RLD to show impurity levels are similar between the two products, or submit toxicology data to qualify the general safety of the impurity. This can be via genetic toxicity assays to determine mutagenicity potential or general toxicity studies. And also an applicant may qualify an impurity as a metabolite. So now I'll describe some common issues or deficiencies that are division issues for impurity, in impurity review. One is the use of in silico methods to qualify the general safety of the impurity. In silico methods are only adequate for bacterial mutagenicity via a QSAR. In silico methods are not validated for the general endpoints of general toxicity. And additionally, using a read-across approach with a surrogate compound is not an adequate approach, meaning using toxicity of a similarly or structured compound to justify the safety of a target compound is not an adequate approach. Also, I mentioned that an applicant may try and qualify an impurity as a metabolite, and this is because ICH Q3A and Q3B state that impurities and degradants that are also significant metabolites detected in animal or human studies are generally considered qualified. However, we have seen scenarios where an applicant may submit published information stating that an impurity is detected in animal or human urine, but there's no quantitative exposure to that impurity as a metabolite. Or there's quantitative systemic exposure, but the level is not sufficiently high, meaning that it's lower than the proposed clinical exposure to the impurity. And then a third pitfall is irrelevant justification, like I mentioned, submitting data for a surrogate. And then another deficiency that we issue sometimes is when an applicant submits proprietary data. Proprietary data cannot be used to justify impurity limits without permission from the owner of that data. The next review type we will cover are extractables and leachables. or ENLs. ENL studies identified compounds that are extracted or may leach from the container closure system, or CCS. And PharmTox reviews safety of ENLs above safety thresholds, like the threshold of toxicological concern, or TTC, or the safety concern threshold, or SCT, both of which are thresholds below which a compound may pose negligible risk of carcinogenicity or other toxic effects. Again, there are different approaches to qualifying ENL. One is to demonstrate that extractables are not present in a leachable study. So extractables are also what we call potential leachables. An extractable assessment is conducting using extraction solvents on materials or packaging or container closure systems, so filters, tubes, caps, etc. And a leachable assessment is performed using actual drug product or a solvent that mimics that drug product. Another approach is to demonstrate that ENLs are below the safety concern threshold that I mentioned. Submitting genetic toxicity assays to predict mutagenicity or general toxicity studies for compounds exceeding these safety thresholds. Or using a surrogate molecule. So when there's no compound-specific tox information available, an applicant may use a surrogate approach as well as quantitative structure-activity relationship predictions. Now here's some common deficiencies we issue with ENL reviews. One is inappropriate calculation of the analytical evaluation threshold, or AET. And this is a threshold above which a compound has to be identified, quantified, and reported for its potential toxicity assessment. Another is when all compounds above the AET or SCT are not properly identified, meaning that a compound name is provided without a structure or vice versa. Classifying ENLs by the Kramer classification method in lieu of submitting a safety assessment is not acceptable. This is because the Kramer decision tree, right, classifies compounds into one of three classes of low, moderate, or high probability of toxicity with using structural elements. And I mentioned that using a surrogate approach may be used for safety assessment of ENLs when compound-specific tox information is not available. But in order to determine if a surrogate is appropriate for an ENL, the ENL in question has to be adequately identified. Next, we will cover excipient reviews in generics. So excipients are ingredients of the formulation that are added to enhance the performance of the drug product but do not exert therapeutic effect. Again, when conducting our reviews, the goal is to ensure that the proposed generic has the same safety profile as the RLD when used according to the labeling. Our initial assessment includes comparison with approved levels with, in similar contexts of use using the IID. Now, if these comparisons cannot justify the proposed use of the excipient, additional assessment is conducted by DPTR on a consult basis. There are different approaches to excipient qualification. One is using or leveraging levels in products with the same excipient in similar contexts of use. Submitting PharmTox data to inform the safety for the route, dose, and duration of the proposed generic product. And also taking into consideration specific patient populations like pediatric patients. So now we'll discuss some common deficiencies with excipient review. One is referencing the IID for excipient use under a different context of use. Another is referencing an unspecified grade of an excipient in the IID for a specified grade of an excipient in the formulation. In such cases, a bridging approach may be used with similar grades of an excipient. Now, when there's alternate routes of administration, not submitting information to qualify the local or systemic safety for those alternate routes like ophthalmic, rectal, buccal, sublingual, dermal, etc., may lead to deficiencies. And then along those same lines, not submitting information for sensitive populations like pediatric patients, particularly newborns and young infants that may have immature metabolism, or patients with conditions that need dosage adjustments like patients with renal or hepatic impairment. Now I'll go over some overall challenges that that we see during our PharmTox evaluations. One is issues with data integrity. For example, suspicious data patterns like data repetition or biologically implausible data. Claims of GLP compliance, meaning that there's a GLP compliance statement, but when we review the study, we realize it's not really GLP compliant. Another one we've seen is the same data reported for different studies, or even the same data reported between different species. We've also had instances of false negative results. These are primarily in vitro studies done to detect bacterial mutagenicity. Sometimes FDA has data to support a positive result, but the applicant or firm submits the negative results, which may raise some questions about data study integrity. Another challenge we see sometimes is missing information in data reports, like missing data, incomplete methods or results, unsigned quality assurance documents, or pathology reports. I've mentioned that submitting literature references may serve as a justification for general toxicity. However, something that we see sometimes is not submitting the full article or the full reference, and that can be advantageous in ensuring that that's an acceptable approach. Missing quantitative data for metabolite levels in plasma of animals or humans. The applicant must demonstrate that quantitative exposure of the metabolite and that it qualifies the impurity levels. Again, referencing proprietary data under another application without right of reference is not an acceptable approach. And lastly, misunderstanding the major versus minor amendment. It's important to know that safety justifications are generally defined as major, and data submitted within the cycle that requires a PharmTox consult may extend your goal date. Next, I'll explain some best practices for successful PharmTox justifications. I mentioned that a major pitfall is not evaluating the impurity, ENL, or excipient within the context of use of your drug product. And I'll describe some best practices for the 3 review types that I covered, impurities, ENLs, and excipients, but some of these apply to all. So for impurities, conduct comparative impurity analysis with the RLD, and then also controlling the impurities at limits that support that analysis. Now, if impurity specifications exceed the threshold of toxicological concern, or TTC, which is an estimate of safe exposure for any mutagenic compound, then a mutagenicity assessment is necessary. And this can be in the form of a QSOR analysis using appropriate models. And if the impurity is predicted to be negative for bacterial mutagenicity, then it can be controlled at QT per ICH Q3B and Q3A. And if the impurity limits exceeds the QT, then general toxicity needs to be addressed. And again, this can be in the form of full literature references and full study reports to justify the safety of a proposed higher level. And also, when the applicant is conducting their own general toxicity study, it's imperative that they consider the context of use. For metabolites, I'll reiterate the best practice is to submit that quantitative information of the metabolite levels in plasma of either animals or humans. For ENLs, I mentioned a surrogate approach may be used for compounds that there's not that compound-specific tox information. And also know that structurally similar ENLs can be grouped together in safety assessment with appropriate justification and identification of the individual extractables and leachables. This can be with a name, structure, and CAS number, and any other relevant information. For excipients, comparison with the RLD, comparison with approved levels for proposed route using the IID, and submitting literature data to justify the safety of the proposed level of the intended, for the intended context of use of your drug product. And next, I will finalize my talk by providing you with some avenues to get advice from OGD. And one is control correspondences, which are a great resource, and there are several inquiries that we can address with the control correspondence. One is related to the inactive ingredients of a formulation, request for Q1, Q2 formulation assessment, determination of the maximum daily dose, determination of thresholds for ENL studies like calculation of AET and SCT, questions pertaining to ICHM7, so treatment direction of the drug product, acceptable intake for mutagenic impurities, and whether or not the drug product is excluded from ICHM7 and falls within another scope like ICHS9 for products with advanced cancer indications. Some aspects of acceptable intakes for nitrosamines, and input on a proposed non-clinical study design. It's also important to note that there are limitations to what can be addressed in a control correspondence. Although we can provide input on a non-clinical study design, a control correspondence is not intended to conduct a full study review, and therefore a safety call is not addressed in a CC. Here's some links to the resources that I covered during my talk, as well as links to DPTR's prior SBIA generic drug form presentations, which also highlight some of the topics I discussed today. And now I will conclude my talk with 2 challenge questions. The first one is, which one of the following is not an acceptable approach to assess general toxicity of an impurity that exceeds QT? Perform comparative impurity analysis with the RLD, use a read-across approach with a surrogate compound, cite published scientific studies demonstrating the impurity is a metabolite of the drug in vivo at levels that exceed the proposed maximum exposure, or D, perform a repeated dose toxicity study in rats, taking into consideration context of use. Anyone? Yes, it's B, use a read-across approach with a surrogate compound. I mentioned that that is an acceptable approach for an ENL, but not for an impurity. Challenge question 2, OGD farm tox does not address which of the following inquiries in a control correspondence? A, questions pertaining to ICHM7. B, questions regarding a protocol of a proposed non-clinical study. C, determination of the AET and/or SCT for extractables and leachables. Or D, review of genetic toxicology study report to characterize genotoxicity prior to ANDA review. D. Again, a control correspondence may address or provide input on a non-clinical study design, but it does not review the study itself. And with that, I'd like to thank you, and I look forward to answering any questions during the panel discussion.
Thank you very much to all our experts on previous session. We invite all of our panelists to come up and have a seat, please. And we will start in-house like we did yesterday. We will start at the mics. If there are no questions in-house, we will go and take as many as we can from the online repository that we have. All right, which one came first? Okay. On the far side, please. Question in-house.
Yes, hi. My question is for Dr. BE. When you were presenting GDSA-BE, I think that was terrific. It's great. It's going to provide a uniform approach. My question is, how closely integrated is that with the current PSGs as they were formed over, to say, the past 20, 25 years? And is the data that is going to be taken from the GSD, from that system database, is it actually integrated with how those were formed today and in the past 15 years? So my concern would be that the threshold of burden to obtain approval for an ANDA is higher than the threshold that the NDA had from 10 or 15 years ago. So is that true, or is there a mechanism in place to make sure that that doesn't happen?
So first, thank you for the question, and I'm very proud that you can pronounce GDSA-BE right. And second, I'm trying to understand what you're trying to alluding to. So I think you're referring to the legacy data.
Yeah. So if you have this data, right?
Right.
That was used to form the PSG, presumably, right? How is the current data in the system? Are we using new data from the literature to form PSGs, modern day PSGs, that might not have been needed by the NDA holder themselves? For example, IVPT testing was not something performed earlier by the NDA holders, right? So Now it's a requirement, right? Which is fine, makes sense, right? But how much of that is going to be newer data, right? Sometimes the NDA holder, if the NDA holder did not have to perform some of those tests and have some of that data, because you can see this by reviewing the SBOAs, that the data did not exist in some cases, right? So, and they might be discontinued, right? So there's no way to even get their product to determine if it's similar. So unless you obviously use the RS, the current RS. But is there data that's needed today that was not, didn't exist before? So in other words, you're going to need to get approval, you're going to have to meet the PSG requirements, but the NDA holder themselves did not meet those requirements. So in other words, the threshold is higher now for the generic to get approval.
I wouldn't say the threshold is higher. By the way, my answer probably is not going to be fully addressing your question because GDSA or GDSA-BE is actually a generic drug structure assessment tool for bioequivalence reviewers. And for PSD that you're alluding to, that's another office, right, ORS. And currently, ORS is not using GDSA system for their PSD development yet. But you're right, and we are trying to incorporate some of the legacy data in our GDSA-BE system to inform our bioequivalence assessors to make informed decision. But that data is based on the bioequivalence assessments in the past years, right? Like the deficiency language that I was alluding to in my talk, and also any critical decision-making process during the past year's bioequivalence assessments. Of course, for those discussions, ORS or PSG development offices, they are also involved in those discussions too. So I wouldn't specifically address, how this GDSA-BE tool will help, ORS offices in terms of the PSG development, because that's a future state if there is. I hope that answers some level of your questions. Thank you. Thank you for the question. Again, I'm so proud that you can pronounce GDSA-BE tool right.
Thank you.
Thank you.
Question here, please.
Yeah, this question is for Ang on the pediatric excipient levels. Is the agency going to provide us a resource to know what are the acceptable levels for pediatric use for the excipients, like providing an additional column in the IAD database?
Yeah, thank you very much for the question. I think, as I mentioned in my slides, at this very moment, there's no extra column in the IAD specifically designed for the levels approved for different age groups in pediatric population. I cannot give you like a conclusion or a foreseen date that when this data will be or if it will be added to the IAD at all. But I can only say at this very moment, if there's any question or proposal or concern about the safety for a specific excipient used in the test formulation, then the applicant is more than welcome to send a control correspondence to confirm with the agency before the ANDA was submitted. So that's my question, my answer to your question. Hopefully it helps a little.
Okay, thank you. So can I ask?
Let's go to online, please. Online?
First question for either Tao or Yang. Is excipient level critical in the drug approval process?
Are you saying if excipient level is critical for ANDA approval?
Is excipient level critical in the drug approval process?
So, Tao can probably chime in later, but to me, the acceptability of a proposed excipient level, it is a critical aspect during our ANDA review. So the excipient level, as I said, it needs to be demonstrated to not affecting the efficacy or the safety of the test product or generic product comparing to the RLD. So yes, I would say that is a critical aspects in our ANDA review. And Tao, if you can.
Sure, I would say everything that we require you to submit is critical. If you have any additional questions, then please go ahead.
Yeah, This question is to Tao only. It's great to know about the GDSA-BE tool. Is this going to affect us the way we are submitting the data in future on the BE data?
Sorry, say that again. It will.
So is there any effect on us the way we are presenting the data, bioequivalence data in the application? This GDSA-BE tool is going to affect us on the way we present the data to the division.
Right, you get that. You get that point, and only mentioned once in one of my slides. Really, that is the structured data submission, right, from the end applicants. You're right. In the future, we are planning to have structured data submission, but right now it is currently under development for the requirements. So more details will be released later.
Like we are presenting currently, structured product labeling now. So labeling is a structured one already now.
Exactly. GDSA is the generic drug structure assessment for the entire generic drug program, and labeling is actually the first stage. And BE is the second stage. You're gonna hear more and more programs, releases in GDSA-BE, there'll be safety, filing, later on in future days. So you're right, labeling is already in structure stage. Yeah.
Thank you.
Thank you.
Thank you. Let's go to the far mic, please.
Good morning, all. My question was...
You need to stand close up, please, so we can hear you. Also, the people online.
Yeah. Good morning, all. My question was similar to previous questionnaire for Speaker Tao regarding the GDSA labeling. So I'm gonna switch my question to Speaker Yang for history. So we saw that all of the innovations and discoveries happened only after the tragedies, like innovations followed by the tragedies, being it thalidomide tragedy, being it Tylenol tragedy, and everything. So do you think FDA is providing any preventive measures at the back end to streamline or cut the future tragedies to be happened?
So this is a little bit history talk. So about the example, the tragedy, the 1937 tragedy I was talking about in my presentation, I was not in the right position. I believe I'm not in the right position to judge what is the best approach. I can only say, as you can see, the tragedy occurs in the autumn in 1937, and the law was passed in 1938. So only after 1 year, at that time, I don't think it's the Congress actually made a very, very fast move. Actually, after the incident was firstly reported, thousands of inspectors were sent across the country to collect data, to visit patients, to visit victims, and try to gather as much information as possible to do that. So I would say this is very, very unfortunate. As you mentioned, there are actually more than one tragedy in the history of drug development, but I can only say anything that we see that either in the laws or in the guidances, it is a little bit later or a little bit delayed comparing to what if, God forbid, what anything bad could happen. So we can only do either call that a corrective action or a preventive action after something bad happened. But I think the one thing that I do want to point out is, a quick action is always needed, especially for bigger issues like what you mentioned and what I had in my slide. So I'm not sure if that answers your question, but I will only say anything that either the FDA or the Congress pass as a law to prevent something bad or something similar bad happen is always a little bit delayed. It cannot prevent any bad thing to happen in the future because we don't know what's going to happen. But yeah, I think that's my point of view.
Thank you for your answer.
So if I could add a little bit, I just want to say GDSA-BE could be the tool, right? Yeah. Now with GDSA-BE, we are able to make more consistent, informed decisions. So that could really help us prevent any future tragedy to happen.
Correct.
Thank you.
Yeah, as I understood the relation between the BE and labeling from Speaker Shabnam's presentation, I had the similar question for that as well.
Absolutely. Thank you.
Thank you.
Thank you. Let's go online, please.
question for Yang. Does FDA have plans to add pediatric information to the IID? If yes, what time frame? And if an applicant can't find strong supporting information from the public domain, can they submit controlled correspondence to OGD for input?
Yeah, thank you for the online question. I think that's very similar to the question I addressed before, but let me just reiterate what I think is true. At this moment, there's no a specific timeline for adding additional pediatric IAG safety data into the IID. There might be additional data in the future, but unfortunately, I cannot give, and I'm not in the right position to give a clear timeline. But in that question, that's right on the spot. If there's any question or any new proposal from the applicant side, always a good idea is to confirm with the agency through a control correspondent. There's one thing I didn't say in my previously addressing the question, similar question is, because just be considerate that the response time for a controlled correspondence is usually kind of fast turnaround. So it would be very helpful for we reviewers to review the safety acceptability for excipient if there's additional data, literature including, to be submitted alongside with the control correspondent. That will kind of speed up or expedite the review cycle for control correspondence for this kind of question.
Thank you. Let's go to the mic right here in front of me, please.
Thank you. Hello, and thank you for all the presentations. My question is for Dr. Dancy. For an extractable impurity that has been detected above the AET, but has been identified and evaluated as safe, for example, it's well above or below the PDE at the amount detected, can the agency please confirm or clarify if that extractable still needs to be included in a leachable study?
Yes, any extractable above the AET needs to be identified, quantified, for us to determine whether or not it needs further evaluation, right? And we have these safety thresholds on top of the AET to determine whether or not it's potentially mutagenic or there's any other general toxicity.
Okay, thank you.
Thank you. At the mic at the far end, please. Step up closer, please.
Yes. Good morning, and thank you all for your presentations. Very informative. This is probably for Dr. Dancy, but whoever would like to answer, feel free. So if there's a general category for an excipient, I'm going to use the example of microcrystalline cellulose, but there's also specific ones for given grades, like 102 or 202. Can the applicant justify using the general category in the IID if it has a higher limit for the with all the other things, that it's the right route of administration, all of those things? Specifically, can we provide that justification? Do we need to do a CC where here, like, the grade differences are related to particle size and bulk density, but not related to something else?
Yeah, so I'll reiterate that if there's any question regarding the excipient, like a control correspondence, as you know, my colleagues have mentioned, it's a great approach. And then when it comes to your question, I mean, normally we're a consult-based division, right? So normally, like, OB will do their initial assessment, and then if there's any question, you know, if the excipient cannot be justified based on the IID or approved products, then they'll consult us regarding that specific grade, and we'll conduct our evaluation. And I also mentioned that during excipients, we do use a bridging approach a lot of the time. But again, that's once it comes to us, we do have to conduct that general evaluation on the that specific grade. Yeah.
Okay, thank you.
Thank you. Online question, please.
Great. Question for Shabnam. How do we perform comparative analyses when the RLD is discontinued or unavailable?
Hi, thank you for your question. Indeed, when the RLD is unavailable or discontinued, we recommend that you reach out to us. And there are two venues to do that. There is a venue of controlled correspondences, and there's also the venue of FOIA. If they, for instance, the RLD is so old that is hard to find, but we definitely recommend that you reach out to us and find a solution, because the generic has to be compared to its RLD. Thank you.
Thank you. In-house, first year.
Yeah, I have a question regarding the Bio-IND procedural related question. So we understand that once a Bio-IND is submitted, agency has up to 30 days to respond if there are any clinical hold comments. But once an applicant responds to the clinical hold comments, how long does agency take to clear the protocol? I mean, is it 30 days again, or is it longer?
I don't think that 30-day hold is there for the clinical hold. I think that 30 days is for the initial response. We try to expedite it as much as we can.
Yeah, I think the guidance for INDs basically indicates that response to the clinical hold comments are also responded within 30-day time frame. But for Bio-IND, it's not very clear.
Sorry, can you say that one more time?
For the INDs that we submit for the NDAs and all, the guidance states that even response to the clinical holds are responded within 30 days. But for Bio-IND, like, there's no such clarity.
I would have to verify that.
Okay, thanks.
Thank you.
Can I ask one more question?
Go ahead.
This is for Dr. Dancy. A very helpful presentation on the extractable impurity limits and the other parts that you covered. So you mentioned that for impurity limits, proprietary data for RLD cannot be like referenced to justify the limits. But if the applicant carries the analysis and generates data with the RLD samples, would that still be considered proprietary information?
I think that would be considered a comparative impurity analysis, right? So that would not fall, I don't think, under proprietary data, as long as the comparative impurity analysis is conducted using validated methods. And for that, we normally collaborate with OPQ to determine whether or not the comparative impurity analysis is adequate.
Okay, thank you.
Thank you. Question at the far mic, please.
Okay, thank you for your presentation. And this question is more to Dr. Liu, but maybe a little bit with Dr. Nancy also. So it's very informative about that the excipients level is addressed to different populations, especially pediatric. But there are cases during like generic development or that even to submission stage that the ROD have not like fully complete their assessment around pediatric, or that it's further extended by like new indication. Like when will this evaluation extend to a different population be triggered? Like when there is a potential, or effectively when that ROD have this indication, and or that will this may result in generic having a different population or indication than the ROD on labeling?
So let me make sure I get the question. You're saying if a generic drug referencing an ROD and the ROD label has not covered, for example, pediatric patient yet, but it may be expanded to that population, what should be evaluated during the generic? Is that
Exactly.
Okay, so that's more relating to the drug label revising, or apparently additional data will be needed. So I would say if what the current labeling, the current RANG labeling is specified, that the drug is not indicated to pediatric patients when the ANDA is submitted, then at least at this, I think at the OGD, the current practice, if the current drug labeling does not specify pediatric use, we will not concern about the safety in pediatric. But if there's any evidence or any record showing the NDA drug or the innovator drug is actually considering to be given to a pediatric patient down the road in the future, and we will take that into consider as well in our ANDA review. But to your question, we pretty much stay with the current ROD label.
And just to clarify, is there any case that any chance that it will result in different population, like maybe that the ROD will have this extension to pediatric, but the generic do not have?
First of all, I would kind of ask you a question. How it was hypothesized that an ROD will be given to pediatric patients if the current ROD label doesn't say so. So as a generic drug company, that's actually really my question is, we are the generic drug is trying to be pretty much exactly the same as the RLD product, right? And one of the key important information or the most important information will be gathered or specified in the current RLD label. So what triggers that, we have the concern that this drug will be, like 5 years later, will be used in pediatric patients. That's, so the same thing for regulators, for reviewers, we do the same thing. At this very moment, when we're reviewing it and, we just follow what the RLD label is talking about, especially for like the target population. I can, hopefully this answer a little, but maybe others can chime in.
Perhaps we can go to an online question, and you guys are here, and maybe you can get to the speakers for a lengthier explanation during the break. Okay?
Thank you.
Online question, please.
Question for Andrea. A BE study that was previously submitted under a Bio-IND needs to be performed again with minor changes to the study protocol. Do I need to submit a Bio-IND amendment?
Yeah, if you're going to change, if you are going to make changes to the protocol for that study, then a Bio-IND amendment does need to be submitted.
Thank you. Mike, closer to me, please.
Yeah, so thank you. I don't have a question, but I'm Partha Roy, the Office Director for the Office of Bioequivalence. I just wanted to address the question which gentleman has regarding the PSG and the GDSA-BE. I think there is no difference between what we are doing today and when we are using the GDSA. It's just a tool to make it, the review quicker. Also, accessing a lot of the databases that we have. We are trying to build the system so that we can get to those databases quickly. But the PSG, the information that comes, and that's a regular process, which is coming from our research or coming from NDA, coming from all of the ANDAs that we are reviewing. So there is no change in that process. It's just we are trying to do it much faster, more optimized, and hopefully, ultimately our responses will be more standardized. You find less consistent, more consistent. And also, we're trying to, probably prevent the next tragedy. The data security is a very important aspect for the agency. The agency, people around the world are trying to get to the agency information in the database. So this is another opportunity to put it in the cloud system and have the, the greater security of our data. So that's one of the reasons also we are going into more of the structured review and all that. So, but we can, I don't want to take too much time. But
no, next time you have an answer, put your hand up. Don't come to the line. All right, okay. Give me a mic. Thank you very much, Dr. Roy. Mike at the far end, please.
So this is for the tablet dosage form where RLD has been discontinued and it does not have any scoring on that. But can generic work to have a scoring on the generic tablet? What could be the bio perspective or how FDA take this change? Should I come again? This is related to bio perspective mostly. So there is a RLD which has been discontinued and it does not have any scoring. So as a generic applicant, can they do scoring on the generic tablet? And how FDA take this change?
I can attempt to answer that from a clinical perspective. So if you're proposing a scoring that was not available in the RLD, we're going to go back and evaluate it based on the label of the RLD. So from a clinical perspective, the allowance of that scoring will be based on that. And if, for example, as you score it, if the scores still you can apply the same labeling for the end users to take it, and if that's applicable to that or not. From a bioequivalence perspective, I guess you guys can comment.
Okay, thank you. No more answers from the panel for this one, right?
I would just quickly chime in, just my personal perspective. If the RLD, no matter is discontinued or is still on the market, if it's not scored, then the generic drug, it wants to put the score on the tablet, then needs to consider whether there has been any tested, when the ROD was being approved or being tested or approved or reviewed, is there any concern about keeping the tablet, for example, keeping it intact? Or can that be split or crushed? So that also is a consideration. I mean, it's a more complex question, not just simply say, hey, there's already no reference there, so we can do what we think is the best way to manufacture the generic. I think there's more thinking behind that.
Okay, thank you.
Okay, let's take one question from online, then we'll finish up these two here. We're running a little late, so let's take one from online.
Question for Jimena. How to challenge the feedback received from FDA if a sponsor found it not appropriate? Or how would you
Can you
challenge?
repeat the question,
Sure.
please? To whom was it addressed?
To Jimena. How would you challenge the feedback received from FDA if a sponsor found it not appropriate?
I mean, I guess it would depend on the or what the deficiency was. But if there's any clarification questions, I guess my first thought would be submit a control correspondence. Yeah.
Okay, thank you. Microphone in front of me, please.
Hi, my question is related to GDSA-BE. What's the timeline of its implementation? Are you implementing to a part of the review right now? What stage it is, and is it going to be depending on the type of ANDA submissions? Like, is it implemented all across right now?
So first of all, GDSA-BE is the assessment tool for the bioequivalence assessors, right? I have alluded to a little bit about the structured data submission in the future. Right now, it's currently under consideration. Detailed requirements will come up. So right now, the scope for GDSA-BE, like I said, only about 60 ANDAs have reviewed by GDSA-BE system. So we are taking the approach from oral solid dosage form and to expand to other dosage forms. Right now, we're only focusing on certain study designs. Yep.
Thank you.
Thank you.
Thank you. Last question at the far end, please.
Yeah, thanks. Back to pediatric excipients again, and feeding into the previous question and the question from the panel. So the way a generic manufacturer knows that an NDA is going to be pursuing or have to pursue approval in a pediatric population is because of their PMRs. That's all posted on FDA's letters. So when you see in an FDA letter that they have post-marketing requirements for multiple PED assessments, one can imagine that in the not too distant future, they're very likely to be approved in the pediatric population. So I think part of the question, it was a question I shared as well. If somebody submits an ANDA now and the RLD is labeled only for use in an adult population, and we get an ANDA that's approved based on an excipient level, but later on, it ends up being labeled for use in pediatric populations, and all of a sudden an approved application has an excipient that has a pediatric, some sort of pediatric issue. What's the agency's position on that? You would certainly be applying a standard of pediatric excipient evaluation to any pending or future approvals. What would you do for a consistency standpoint with approvals that have already taken place?
Yeah, thank you. Thank you for the clarification and the follow-up. I think that's a very, very good point, first of all. And also, I think whenever that scenario happened and the drug actually, the PSG, not the PSG, the ROD will be expanded for pediatric use. If that's the case, I think for a generic drug which has been approved, referencing that particular ROD, also needs to have, additional evaluation. For example, the revising of the labeling and everything. I think during that process, at this moment, personally, I have not encountered with this kind of review yet, but that's a very good point, and we can definitely have further discussion back in the office. But I think during that phase, when the approved generic drug also needs to submit like a post-approval revision of the generic labeling, in that phrase, when it considers the use in pediatric patients specifically, additional evaluation within the office will be conducted, and there will be a close communication with the applicant. I think that's the current thinking at least from my personal perspective. But again, I have not encountered into that scenario at this moment. Thank you. That's a great point.